So it's a lot of content. So a lot of students ask me how should they focus? What should they focus on? So let's go through what the exam looks like, okay? So for the lecture exam, there will be 100 questions. Of the 100 questions, 50 of them are going to be on the old materials, which means things from exam 1, 2, and 3, all combined into those 50 questions, which means, though, that these questions are not going to be as detailed as you saw them in the actual exams. They're going to, for example, I have to cover from the intro all the way to digestion, all within 50 questions, which means that there's about 3 to 5 questions per chapter. So if there was a chapter that you really struggled on, if I were a student, what I would do is actually I would focus on the new materials first. And the reason for that is because the new materials consist of the urinary system, the endocrine, and the female and male reproductive, right? So that's 4 chapters. And of those 4 chapters, they're going to be 50 questions are going to be divided into those 4 chapters, which means there's going to be about 10 to 12 questions per chapter. And that means that they're going to be really detailed. So to get kind of the biggest bang for your buck in terms of what you should focus on, if I were a student, I would focus on the new materials because they consist of half of the exam, where all of the previous exam materials make up the other half. Questions on that?
Yeah, so start from the urinary and work our way back to the reproductive, you're saying?
So you want to do urinary, endocrine, male, and female reproductive first.
Okay.
Yep. So if I were a student, and you can study any way you want to, whatever makes sense to you. But if I were a student, what I would do is I would cover the urinary, endocrine, male and female first. And the reason for that is because it makes up half of the exam with just those 4 chapters. So those questions. Yep, go ahead.
So is the lab going to be that way too, or how is that?
Yes, ma'am. Yep, okay. Exactly the same. So the lab has 50 questions. 25 of them are going to be on the old material, and 25 is going to be on the new material. Okay. So there's definitely more questions on the new material than there are questions on each chapter of the old materials. And in addition to that, you should start to find that as you review your old materials, it's a lot easier this time around because you've already had it and the content in this class builds on top of each other. So it makes more sense at this point.
Okay, so Professor Vu.
Yeah.
In that case, never mind about muscle metabolism. I'd rather go over oogenesis.
Okay.
That's a little confusing for me, and I guess we'll have to know that.
Yes, and there's going to be a significant number of questions on oogenesis.
Okay, cool.
Yeah, that's definitely a better bang for your buck. Yep.
Yeah.
Mm-hmm.
I'm like, okay, we don't have to backtrack so far.
Okay, thank you.
Yeah, I figured that would be more comforting for you to hear. So that, so that you know that there's only going to be about 3 to 5 questions on each old chapter. So let's say that there was a chapter that you really struggled on, rather than spending a ton of time studying that, I would actually study the new materials to make sure that I got that content more solidly because more questions are going to come out of them, and just kind of say, okay, out of 100 points, I'm okay losing 5 points for something that, I'd rather take that time and get to the 7 to 10 question pointers. Mm-hmm. Make sense?
Yep.
Okay.
Yes.
All right. So for the lecture exam, you have 100 minutes to take that unless you have extended test taking time. Please, please, please verify that the extended test taking time has been entered correctly. If not, contact me right away. And for the lab exam, there are 50 questions, 20 on the old materials, 25 on the old materials, 25 on the new. You have 75 minutes to do that. As always, spelling counts, particularly on this exam and probably the last exam as well. Oftentimes you tend to have pairs of something. And if you have pairs of a structure, you have to make sure you include right or left. Also, if it's a blood vessel, I noticed that some of the students, only refer to like the pulmonary blood vessel. Well, what type of blood vessel it is. So you want to make sure you include whether it's an artery or a vein. Let me go back and admit. Okay. All right. Questions?
Nope, that's good.
Okay. So you should all be kind of like a pro at this, because if you think about it, it's like a fourth exam. So you've had exam 1, 2, 3. So you have a fourth exam, which is 50 questions on the current content. And then you have kind of a comprehensive section, which is 50 questions on everything else. Okay. The policies, the rules are still the same. You are expected to use Respondus Lockdown with the webcam. Make sure that you're doing a proper environmental scan. And don't wait last minute. The nice thing about this term is that we adopted a new calendar schedule. So instead of only having Friday, Saturday, Sunday to take the exam, you actually have the following week. So you have a little bit more time to study for your exam than the previous students in previous terms because of the difference in calendar. Okay. So make sure you don't wait until the last minute to take your exam, because at that point, it's very hard to give you the help that you need. So you have several days, a larger range in which you are able to take your final exam. So please don't wait until the last minute. And there are absolutely no extensions on them unless it's extremely, extremely extenuating circumstances, like, you were in the ER or you had major health concerns. Because it's not like the other exams where we had more class time for you to have that extension, because when the class ends, when the final day for you to take the exam occurs. So once the class ends, even if I wanted to give you an extension, I can't, unless you have to apply for it through official forms and go through the dean and everything. Okay, so make sure that you give yourself ample time to take the exam. Any questions? All right. So the final exam really focuses on the urinary, endocrine, and reproductive. And that's, as a student, that's what I would recommend you to focus on. And then, of course, as you're doing that, review all the old materials. Okay.
But is it going to start in order from the beginning, or it's going to work its way backwards, or no order this go round?
No, that's a great question. I'm not here to trick students. So I still lump the questions together based on chapters, and you're going to see the new materials first. So
Okay.
the first 50 questions, the first 50 questions will be on the old materials. And then the last 50 questions is like a fourth exam, where 40 of those questions are multiple choice and 10 are fill-in.
Okay.
Yep. So the first 50 are completely multiple choice.
So are the 10 fill-in, are they going to be I'm sorry if you already said this, but are they going to be all on like the last 4 chapters, or are they going to be from everything?
Nope, just the last 4 chapters.
Okay, thank you.
Yeah. These are great questions. Keep them coming. Any other questions? Okay. So we covered what the exams look like, and hopefully I've also given you some strategies as to how to approach them. Go through the new materials first, and as you're going through the new materials, keep reviewing your old materials. A really good source of content to review for your old materials is your previous quizzes and your exams. You should be able to see all of them. Okay, to view, if you've not used that, if you've not reviewed your old exams and old quizzes, make sure you know that you have to use Respondus Lockdown to be able to view them. Okay, use your old study materials. Whatever you've already created, use those. You shouldn't have to create anything new for the old content.
I did try to look at the old exams, and it said on Respondus that a lot of them weren't available anymore. The answers weren't available, so I couldn't see the questions.
The answers are not going to be available after the 2 days.
Right. Okay.
Yep, but you should still be able to see the questions as well as your answer and whether you got them right or wrong.
Oh, I didn't see that, but okay.
Yeah. Go look after the review session. Go ahead and look again. And if you can't see them, please let me know. It
Okay.
might be a setting on my end. Yep.
Okay, cool. Thanks.
Other questions? All right. So knowing that, let me revisit the question I asked at the beginning. What would you like me to cover besides oogenesis? Just oogenesis?
No, I'm looking for, my brain's not working, but I'm trying to find it in my notebook.
How about I start with a general review of everything from the new content and pay particular attention and detail to oogenesis. And then as I'm going through the general review, if you want more details on things, let me know. Does that sound like a good?
Sounds good.
Okay.
Yep, sounds great.
All right. So I'll save the first half because this session is 2 hours long. So I'll save the first half for the urinary and endocrine, and then I'll save the second hour for the reproductive because that's kind of intense. It's like 2 chapters, but like 5 chapters in terms of content. It's very detail oriented. Okay. All right. So starting off with the urinary system, you want to make sure that you're able to list and describe the functions of the kidneys, which includes its ability to filter blood. A nice analogy for the kidney's ability to filter blood, think of it as a washing machine. So you take dirty clothes and you throw it into the washer, right? You don't expect your clothes to be eaten up and disappearing, right? So that means that that washer has to be able to separate the good and the bad, as well as removing the bad content. And that's what your kidneys does. It takes the blood. It doesn't get rid of all your blood, right? It just separates the waste and the toxins from the good stuff that you need in your blood, like nutrients, gases, and other things. And then it removes the bad stuff. At the same time, it has to keep the good stuff. So that's why when we talk about the kidney, we talk about filtration, reabsorption, and secretion. Okay, so that's a main function of the kidneys. Then another function of the kidney is that it has to be able to regulate water volumes. And by regulating water volumes, what does it impact? What does it influence?
Blood pressure and
Yes.
volume.
So blood pressure. And of course, pH levels too, right? Because if it's regulating how much hydrogen ions you keep in your blood, that is going to influence the acidity and basic conditions of your blood. Okay, so blood pressure, pH levels, anything that's related to water volumes, it's going to be able to impact. Okay. All right. Then you should be able to describe the gross anatomy of a kidney. And gross anatomy of a kidney, like if I cut it in half, these are the structures that you can see with your eyes without the need of a microscope. And that's going to include the fibrous capsule, the hilum. Technically, the hilum isn't a structure, right? What's the hilum?
Oh, that's sort of the space where the vessels are entering,
Exactly. So it's a
kind of
space.
curved in spot.
Right. So am I going to be able to ask you that on the lab exam?
No.
I can't because it's not an actual structure,
It's a
right?
space, yeah.
It's a space, exactly. Then you will also want to make sure you know the associated blood vessels, and that's how it goes from arteries to arterioles to capillaries to back to arterioles and then to veins and then to venules and so on. So you want to make sure you understand that transition, okay? You also want to be able to identify the renal cortex as well as the renal medulla and the renal pelvis. And I noticed that on your quizzes that some students are struggling between the difference between the renal pelvis and the calyxes. So you want to make sure that you have a strong definition of how they're different and what happens for them to change the name of that structure, because it's
Yeah,
the same
because
structure.
I struggled with that. I had a question.
Yes, ma'am.
I went
Mhm.
back and forth, and I was really stuck. But then I remembered that that's where the urine is like stored, but the calyx help to move the urine in the proper direction or something like that.
It's not so much as the direction of the fluid, but the structure itself. So after the renal pyramid, so at the base of the renal pyramid, there is what do you find at the base of each renal pyramid?
The renal column, I can't remember.
The calyx,
Yeah, which one?
the minor.
Yes. So the minor calyx occurs at the end of each renal pyramid. And that's how you know it's minor, because there's only one renal pyramid. Then when you have two or more minor calyxes joining together, that's when you have a major calyx. So a major calyx is when you have two or more minor calyxes joining together. And then when they all join together, when all of the major calyxes join together and make one solid structure, that's when it becomes the renal pelvis and then exits as the ureter. And then, of course, that's the difference between the renal pelvis and the ureter, is the renal pelvis is still part of the kidney, where when the tube exits, it becomes the ureter. Does that make sense? Questions?
Makes sense.
Awesome. Good. Good to hear. All right. So that's the gross anatomy of the kidney. And of course, you need to know, if you have to know the gross anatomy of the kidney, you also have to know what?
Microscopic.
That's right. And the functional unit of the kidney is the nephron, and that's going to be the microscopic. So for the nephron, this is something you can't see with your unaided eye, so you'd have to use a microscope to see it, hence microscopic anatomy. You'll need to be able to identify the renal corpuscle, which includes the glomerulus and the glomerular capsule, the renal tubules, which is the PCT. What does PCT stand for again?
Proximal.
Convoluted tubule.
Beautiful. Great job. Isn't it a shame that you can't take the exam together? You can combine it, and then you'll be able to answer everything. But
Two brains are
that's
better
why,
than one.
exactly. But that's why I recommend study groups, because someone always has something to contribute. Okay. So you have plenty of time. You have this whole week to study together. And actually, if you are able to form study groups, you can get extra credit for that. So just a gentle reminder of that extra credit opportunity. Okay. Remind me though, if I don't get to it, remind me to talk about the extra credit opportunity at the end. Okay.
Okay.
It's the same one. I just want to make sure you have all the parts. All right. So the PCT is the proximal convolute tubule, and then the nephron loop, and then the DCT. And remind me what the DCT stands for again.
Distal convoluted tubule.
Right. And knowing the name actually will help you identify it, right? Because following the glomerular capsule, what's connected directly to the glomerular capsule?
The glomerulus.
Yes. So it's not connected. So you have the glomerulus being surrounded by the glomerular capsule. So it's not technically connected to each other. It's just one surrounds each other. Think of it like a baseball mitt surrounding your hand. Okay.
Oh, okay.
Yeah. Okay. So the glomerular capsule is connected to what? I'll give you a clue. It's one of the three renal tubules.
The PCT.
Yes, and it makes sense because P stands for proximal. So whatever set of tubules is closest to the glomerular capsule, that's why it's called the PCT. So it doesn't matter what it looks like on a test, whether I use this image or another, as long as you follow that connection right after the glomerular capsule, it absolutely has to be what?
The PCT.
Right, because that's why it's called the proximal convoluted tubule. Proximal because it's closest to the glomerular capsule. Convoluted because it's not straight tube, it's like really curvy. And tubule because it's a tube.
Tube, yeah.
Mm-hmm. So following the PCT, what would you find next?
The nephron loop.
Yes, ma'am, that's right. And then, of course, after the nephron loop, you see the
DCT.
right. Makes sense because it's the distal one. It's the set of convoluted tubules that is furthest away from the glomerular capsule. So you don't have to memorize this, right? It makes sense. So use the name to help you identify and be able to correctly name structures, okay? Following the renal tubules, and this one's a little tricky because it's based on whatever textbook you're reading, because some textbooks will say that the collecting duct is part of the nephron, where the one that we chose to use for your class does not. And that's why the renal tubules is separate from the collecting duct, okay? Then within your description, you also want to make sure you know the structure, function, and the tissue lining. You're like, tissue lining, why do I have to know that? Because it helps you understand the function. Remember, one of the things that I want you to leave this class with is that structure and function go hand in hand. The structure determines the function, and likewise, function determines how it is structured. They always go hand in hand, okay? So understanding the tissue lining of the parts of the nephron will help you understand what each part does. Let me give you an example. The PCT, the proximal convolute tubule, is lined with what?
Simple cuboidal.
Yeah, epithelial tissues.
Cilia.
Beautiful. I love your answer. Simple cuboidal epithelium. And with what?
Microvilli.
Microvilli.
You made me so happy. Microvilli, exactly. And
Microvilli.
Have you heard of microvilli before?
Yeah, I remember from another chapter somewhere.
Yes, beautiful. Exactly. What chapter?
Digestion.
That's right.
Okay.
So anything that has microvilli, at this point, you should be connecting the structure microvilli with absorption, right? Because microvilli helps to absorb things that move through. It just happens that in the digestive system, since it's the first time it's being absorbed, it's called absorption. Where when it happens in the kidney, because it's the second time moving into your body system, it's called reabsorption. Okay. Now, it turns out that of the renal tubules, the PCT, the nephron loop, and the DCT, the PCT is lined with the microvilli, where the nephron loop and the DCT has a lot less to zero microvilli. And that's why the PCT, what happens mostly in the PCT?
Reabsorption.
Yes, right? That's one of the things I made you know, right? That about 60%, or you can just even say a majority of reabsorption occurs where?
And the
In
PCT.
the PCT.
Does it make sense now?
Yes.
Mhm.
Right? So function and structure go hand in hand. Yes, Gina?
So microvilli and cilia are completely different things, right?
Yes, they are. That's a great question. So let's analyze that question. So can someone tell me the difference between cilia and microvilli?
So I guess the microvilli are extensions of the, like, kind of like projections of the cell, maybe?
They're
Protrude.
they're both extensions.
Okay.
Oh, they're both extensions? Okay.
Yeah.
Oh,
But
no,
they look different. They look different.
I just imagine all of them like wiggling around.
Yeah. Yeah, they do. They absolutely do. Yep. So you would have learned in digestive system that cells that have the microvilli increase the surface area, just like the villi, right? And that helps to slow things down, which improve absorption. Where cilia are longer hair-like extensions, Not as long as flagella or other things like that, but it's still longer than the villi and the microvilli, and they actually help to move things. So microvilli doesn't help to move things, it just helps to slow things down so that it can be absorbed. So that's the difference between cilia and microvilli.
So the microvilli are like part of the membrane. Are they?
Yeah,
Okay,
exactly.
got it. So things can
Yep.
go in and out. Okay.
Yep. So let's use another example. So your upper respiratory, completely lined with cilia, right? Because it has the stratified columnar epithelium, right? Which makes sense because it helps to move things along, and that's why when something like debris is stuck in your throat, what do? Right? You hack it out, right? And that's because of the cilia. The cilia helps to do that. Okay. So yes. So cilia, microvilli, very similar, looks similar, but one is longer than the other, and the function is different as well. Okay.
Cool. Thank you.
Yeah. All right. Then you also want to be able to describe the process of the urine formation in detail. You know, that's important to know. You know, there's going to be at least 5 questions on that. Why? Because it's physiology, right? And this class is anatomy and physiology. So we ask you a lot of things about anatomy, but we also want to make sure we ask that you know the physiology, how things work. Okay. And that's what your information is about. So I guarantee you will have at least 3 to 5 questions on your information. It's worth your time studying, and it's detailed. So you want to make sure that you do know the details, okay? Then go through make sure you know the structures after the kidney. So that's going to include the ureter, the urinary bladder, and the urethra. Make sure you know the structure, function, as well as the layers, okay? And then lastly, be able to describe the steps in the micturition process, which is relatively straightforward. Okay. Questions on the urinary system? Okay. All right. So gross anatomy. Let's start with number 1. What's number 1?
Adrenal.
Yeah.
Glands.
Yes. So if this was on your lab exam, you will need to write down adrenal gland, and you will want to also include what?
The side that it's on?
Yes, ma'am.
Left.
Left.
Yep. So this happens to be on your
Left.
yes. Okay. So sometimes I always, you can always have a scrap piece of paper as long as on the exam during your environmental scan, you hold up that scrap piece of paper. It's got to be blank, nothing on it, and show me the front and the back. Okay. One thing that I always do, because, I tend to switch things all the time. Like I can't tell my right from my left. Legit. Like my friends would ask me for directions and I'd be like, I told you to take a left, and I'm pointing to the right. Okay. So I struggle with directions. So what I have to do whenever I take an anatomy test, I actually have to go like this. I just make a line, and then I write down my right is the left because it's anatomical position, and my left is actually the right. So that helps me just to kind of keep clear what's right and what's left. Okay. So that's a good thing to put onto your scrap paper just to remind you, because I have seen some of this, and I know you're probably real mad at yourself. You were able to identify it, but you switched right and left. Okay. All right. Number 2.
Kidney.
Left.
Left kidney.
Beautiful. That's what I like to hear. Left kidney. Okay. Number 3.
Left ureter.
Left ureter.
Number 4.
Urinary bladder.
Urinary bladder.
Yes, I just had to take half a point off of one of your colleagues, one of your peers, because all they wrote was the bladder.
Aww.
Do you only have one bladder in your body?
No.
No, you don't. You have the gallbladder. You have the urinary bladder. You have, yeah.
Uh-huh.
Yep. So you have so you have to be specific, okay? What's number 5?
Urethra.
Yes. Okay. All right. Number 9. You should know number 9 as well.
Renal, right.
Right.
Renal artery.
Yeah.
Yes. Perfect. And number 8?
Right renal vein.
Right renal vein.
Yes. You are not responsible for number 7 nor number 6. So we'll just take those out. All right. So that was the gross anatomy. Let's move on to microscopic anatomy. What are you seeing right here in this image?
Oh, that's what the Galerius capsule stuff look like.
Yes.
What are they
So-
called?
Yep. That's what I'm asking you. What is this
Oh,
called?
what is called?
That's the nephron.
Yes. Okay.
What is it?
Nephron?
Yes.
That's
Oh yeah, duh.
No, there's no dir. This is the best time to have those dir moments. Okay. So this is the nephron. Yep.
Yes.
So a sample test question in the past is, what are the functional units of a kidney? What would you say?
nephron.
Nephron.
That's right. Okay. So this is the nephron. So we know that the nephron essentially has 2 main parts. What are the 2 main parts of a nephron?
Mhm.
The proximal and distal.
Ooh, no, because it does have the proximal and the distal. It also has the nephron loop, but all together, those 3 parts make up what?
The nephron loop?
No?
No, that's just the part. Oh,
Mm-hmm.
The ascending and descending.
Nope. Now you went into even deeper detail about the nephron loop, which is good.
Is it the renal capsule and uh-oh.
Wait, the collecting duct isn't
I don't
considered
know.
part, right?
Nope, it's not. That's right.
And the renal tubules?
Yes, you got it.
Oh,
That's
yeah.
it.
Mhm.
So the 2 main parts of a nephron is the renal corpuscle.
Oh, corpuscle.
Oops, let me just spell that right. And the renal tubules. Okay, so the renal corpuscle includes these 2 parts. Let's do 1 here and 2 here. So what's number 1?
Afferent.
Afferent.
In general. I'm not looking for the specifics yet. So I just want the general.
Arterials?
Just generals. But you're right. So it starts off as the renal artery, becomes the afferent arterial, then what?
Oh,
Then the the
Yeah,
glomerulus?
that's it. So the renal corpuscle includes the glomerulus, which is the
Oh,
blood
the
vessel.
glomerulus capsule.
Yes. So the glomerulus is a network of capillaries. And then number 2 is the glomerular capsule. The glomerular capsule, in old times, if you studied this when I did, we used to call it the Bowman's capsule. But please don't use Bowman's capsule, right? Because the terminology that I want you to use is the most common one. And that's why we're really pushing you to use the terminology that's provided for you on the PDF, because it's the most common, most current terms. Okay. All right, so that's the renal corpuscle. That's the first part. Then the second part is the renal tubules. Remind me what are 3 parts of the renal tubules again?
PCT, nephron
PCT,
loop, and
nephron loop, and
DCT.
DCT.
Perfect. Excellent. Okay. And then of course, not really a part of the nephron, according to your textbook, is this part right here, which is the,
Collecting duct?
yes,
Oh.
ma'am. You got it.
Okay.
Yep. All right. Questions? So now you're going to want to go back into these and add, what's the lining? What are they lined with? How does the lining connect to the function of each part? And how is the function connected to your information? Because they are all connected. Okay? All right. So, because I want to save some time for the endo. This is my favorite picture. It's lovely. It really does a good job of showing you everything. Right? So what we just covered in the previous slide is just the nephron. Connected to the nephron, or associated with it, are the blood vessels. Okay, so do you remember one of the objectives that you need to know for the urinary system is to be able to take a look at all the associated blood vessels? Well, this is it. This is all the associated blood vessels plus the nephron itself. Okay, so you can see you'll need to know the transitioning of how the blood vessel changes and becomes what, and then moves where, and how that's connected to the nephron itself. Okay, and that's why this picture is beautiful, because it covers the nephron, it covers the associated blood vessels, and it covers your information. Someone just list the steps of your information for me.
The filtration, glomerular filtration,
Yep.
and then tubular reabsorption, and
Yes.
then tubular secretion.
Beautiful. Okay, so let's identify that a little bit more. We're going to start with the glomerular filtration, and look at its name, glomerular filtration. 2 bits of information in just those 2 terms. Glomerular, because it tells you what's the location.
The glomerulus.
It actually occurs between 2 locations.
Oh, and the capsule.
Yeah, between the glomerulus and the glomerular capsule. Hence, glomerular filtration. So where does filtration occur?
Across the blood vessel. Single. Oh, what's it called?
Is it in the glomerulus?
Yep, between the glomerulus and the glomerular capsule. And that's why another way of saying that is that filtration occurs at the renal corpuscle. Because that's the definition of a renal corpuscle, right? The glomerulus and the glomerular capsule. Remember that? Or corpuscle. I have to write that slow, otherwise I spell it wrong. Okay, renal corpuscle. So between the glomerulus and glomerular capsule, which tells you that what was the fluid before filtration and what has the fluid become after filtration. Let's start with before. What was the fluid called when it was in the glomerulus?
Plasma?
Yes.
What was it called? Oh,
Plasma. Yep.
plasma. Okay.
Yep. And after filtration, what has the fluid become?
Filtrate.
Beautiful, filtrate. And this is, or has been in the past, a test question. Okay. So infiltration, which occurs at the renal corpuscle, aka it happens between the glomerulus to the glomerular capsule, it was plasma, and then it becomes filtrate. Okay? Does everything in blood get filtered?
No.
It doesn't. What mainly gets filtered and what should not be filtered?
The components of blood don't get filtered. Like the red blood cells.
There you go. So correct. So I'm going to repeat to you what you said to me. The components of blood are not filtered.
Oh, what's the other word for it? Fix the elements, something.
There you go.
RBCs, white blood cells, and
You corrected yourself beautifully.
okay. But
So
what
this-
about
Yeah, go ahead.
the platelets? They're too big to fit through, right?
So sometimes they can make their way through because they're not actual cells, right? Platelets are fragments of cells. So sometimes they make their way through and sometimes they don't. But mostly what we know for sure, things that cannot or should not move through are red blood cells and white blood cells and proteins. So yeah, I would say platelets and proteins are things that shouldn't make its way through either. But for sure blood, right? Red blood cells. So if you see red blood cells in urine, that's bad. It means you've got some kidney damage or some infection that's causing leaking of the red blood cells into urine. Okay. All right. So that's that's filtration. So you want to be able to know what exactly is filtered. And that's why I love this picture because look, it's all listed here. What is filtered? Okay. All right. And let's see what else. Oh, you want to make sure you know what GFR is. What does GFR stand for?
Glomerular filtration rate.
Yeah. Some of you may be interested in doing that for your final extra credit, which is on the inequities in anatomy and physiology, because even though we try to be objective in science, it doesn't work out that way. Sometimes what we say in the name of science ends up hurting certain populations like GFR. So for some weird reason, we ended up with two rates, glomerular filtrate rate and African American glomerular filtrate rate, because someone assumed that because African Americans tend to be bulkier, their GFR rates are different than other populations' GFR, which is not true, right? So because of that, what happens is that particularly when kidney issues occur, now kidney disease is determined by GFR rates, right? By GFR. So in African Americans, what allows them to be diagnosed as kidney disease is a lot higher, or the threshold is a lot more than in the other populations. And because of that, sometimes they do not get diagnosed for kidney disease, even though they have it. So that's an example of how that can lead to, inequities in care. All right.
I always wondered about that. That's
Yeah.
interesting. So it's just random. They just decided that there was not
Well,
good science behind it.
It's not good science, exactly. That's a good way of saying that. Yep, just based on the assumption that African Americans have more bulk in their muscles and thus their GFR rates are different. Yeah. Yep. All right. So you want to make sure you know what it's being filtered, what's GFR, what's the unit of measurement for that. So all that cool information that I provide in my lectures for you, you want to make sure you know that. All right. Then the second step is tubular secretion. Again, it's telling you what it is, where it happens, right? Where does it happen?
Tubular secretion in the PCT.
Well, PCT is one option because that is a part of the renal tubules. What's another two options?
Is it in the PCT?
It could be. That is the correct answer, but it could also be where else?
The loop of
Is it in the neck?
Yep, it could be in the nephron loop, and it could also be where?
In the DCT too?
Yes, it can.
Oh, man,
Absolutely.
everywhere, huh?
That's why it's called tubular reabsorption, because it happens anywhere along the renal tubules.
Oh, okay. Makes sense.
Yeah. Okay. So just like filtration, we identified where it started and where it ends up. So in tubular reabsorption, where does it start and where does it end up? We know where it starts. You gave me all the answers, right? You said things like PCT, nephron loop, and DCT. So essentially, the renal tubules. So after reabsorption, where does it go?
Into the paratubular capillaries.
Yes. Yes. Don't doubt yourself. You're right. Paratubular capillaries. Okay. So let's take a look at this. First, I want you to see where paratubular capillaries come from. So the paratubular capillaries is this right here, these tubes that wrap around the renal tubules. Okay. So it comes from the efferent E arteriole that then comes from the glomerulus that then comes from the afferent as an A arteriole and then the renal artery. So essentially, blood is brought to the kidneys to be cleaned by the renal artery. It becomes smaller, and then at that point, it's called the afferent arteriole. And how I use to remember that is A in afferent is A in arrive. It's arriving. Okay. And then after it arrives, it becomes the glomerulus, at which point it stops being a type of artery, aka the arteriole, and now it becomes what? What blood vessel does it become?
The glomerulus. The capillary.
There you go. So the glomerulus is a network of capillaries. And capillaries are nice because whenever you think of capillaries, what do you automatically associate with the term capillaries?
Some sort of like exchange or
Ah, you just make me so happy tonight. That's exactly right. When you hear capillaries, you think automatically some type of exchange is happening. And that's what those green arrows are showing you. So the green arrows is showing you if it's a capillary structure, it determines its function, which means exchange. So things are moving from the glomerulus, the capillaries, into the glomerular capsule. Aha! We just talked about filtration. Okay? Then, so most of blood gets filtered except for the formed elements like red blood cells, white blood cells, platelets, large proteins. Those continue on, but all the fluid portions get pushed into the glomerular capsule. So it goes from being plasma to being filtrated. Okay? Now, what leaves the glomerulus are those things, right? The formed elements. And it leads to what's called an efferent arteriole. And how I remember that is E in efferent is E in exit. It's exiting. So once it exits, it's going to wrap itself around the renal tubules because you don't want to have to pee all day, right? Your body filters about 150 to 180 liters per day. If you were to actually pee that much out, you'd be on the toilet all day long and you would die after a day, right? So what has to happen?
It has to be stored and reabsorbed.
That's the word I'm looking for. Reabsorption must occur. Ooh, that's too dark. Reabsorption must occur. Oh, and now it's not working anymore. I messed it up. Darsh. Okay, it's not working anymore. All right. So reabsorption must occur. So that's why the blood vessel changes from being arterial to what? Because reabsorption means something is being exchanged, and in order for exchange to happen, you have to have what type of blood vessel?
Capillaries.
Yeah. And these capillaries happen to wrap themselves around the tubes. What's the term for around?
Peri.
Yep. Peritubular capillaries. That's where they get the name from. Okay. All right. So in tubular reabsorption, the fluid is going from the renal tubules anywhere along PCT, nephron loop, DCT, into the peritubular capillaries. So what was it? What was the name of the fluid before tubular reabsorption? And what has it become?
Before it was plasma, then it was filtrate, and now
Plasma.
Well, I'm sorry, I didn't catch that.
Plasma. Again.
Yes, perfect. So it went from being filtrate. Oh, it went from being filtrate back to being plasma, which is good. Because remember, if you throw your dirty clothes into the laundry, you expect clean clothes back, right? You don't expect half a pants or half a shirt, right? So, yeah. So that's tubular reabsorption. Okay. Then, now, filtration occurs when you like have a load of laundry and you put all your clothes in. But gosh darn it, you went back to your room and you saw one dirty sock. What are you going to do? Are you going to have to wait until the next cycle? Or are you going to be like me? What are you going to do?
Throw it in the washer.
You're going to throw it in the last minute, right? And
Yeah.
that's what tubular secretion is. Tubular secretion goes, oh, I still have stuff I need to clean. It's not going to wait until the next load because you're going to be like, no, I'm not waiting. It's just going to throw it in. And that's what tubular secretion is. So along the renal tubules, things can go in the opposite direction as tubular reabsorption. So see, tubular reabsorption goes from the renal tubules to the peritubular capillaries. Well, if you have that last minute thing that you just need to balance out, you can actually make it go the other way. And when you make it go the other way, we call that tubular secretion. Is tubular secretion going to be as effective as filtration?
nope.
No, it's a last minute attempt, right? It's just a last minute way to balance things out. And that's why in this next image, which I also love, it shows you what is being moved. Okay. So this is in your textbook as well. It's in it's figure 23.22. So you can see that in the blue represents the tubular reabsorption, where the pink represents tubular secretion. So you can see that what's being secreted is a lot less than what is actually being filtered initially. Because again, secretion is only like you found a dirty sock and you didn't want to have to wait until the next cycle. Okay? But there is a back and forth happening along the renal tubules where you have mostly reabsorption and some secretion, but essentially it's just to maintain homeostasis. Okay?
So the secretion does happen throughout all the segment of the nephron. Just it's a smaller.
Careful. Is it all the segment of the nephron?
I don't know. That's what I wrote in my notes. I need to know if I need to cross out all or I don't know. I put this process occurs throughout all the segments of the nephron. So I don't know.
What does the class think?
Help me out, guys. I got to fix this note.
I love that you're thinking about it though, by the way.
Maybe everything except for the glomerular capsule. I don't know.
You got
Oh, okay. Yeah, that makes sense.
it. You got it.
Okay.
Yep. And that's what you were thinking, but that's why I'm, I try to correct students when I know you're thinking about the right thing, but the way that you were saying it wasn't quite
Yeah.
right.
Yeah.
Yep. Mm-hmm.
It's better to say it right though, because it makes more sense in my head now.
Exactly. Yep. But you had the right idea though. You were on the right track. Okay. So with the endocrine system, just briefly, you want to make sure you know the functions of the endocrine system, which include three main things: being able to define the hormones and describing what its role is on the endocrine system. Describe the glands that play a role in the endocrine system as well. So what I would do, and you should already have that in the worksheet. It's kind of a cognitive map, a list of the names of the hormones, and I would recommend studying them together in terms of where they're produced / secreted, and then what do they target and what is their action. Okay, and that will get you a long ways. So an example, again, hormones, where they're secreted, their target, and their action. So I would group the following 6 hormones together: growth hormone, follicle stimulating hormone, luteinizing hormone, adrenocorticotropic hormone, prolactin, and thyroid stimulating hormone. Why? Because they're all produced in the anterior pituitary gland. What they target and their actions are different. So for each one, you would identify that. So for example, the growth hormone, sometimes also written as HGH, or the human growth hormone, targets various cells like bone, muscles. Why? Because it stimulates growth. And the growth stimulation can either be in number of cells or the size of a cell. 2, follicle stimulating hormone, FSH for short. These tend to target the sex organs. And if it's in females, it stimulates the maturation of the eggs, where in males, it stimulates sperm production. Number 3, you've got the luteinizing hormone, or LH, abbreviated as LH. These also target the sex organs, and in females, it would lead to ovulation. So that occurs on what day again? Remind me? Usually, what day?
14th day.
Tuesday.
Beautiful. Exactly, the 14th day. Yep. And then in males, it's going to stimulate the testosterone secretion. Okay. So these hormones, even though we tend to associate them with one gender, they're actually produced in both, and they do have functions in both. Then you have the adrenocorticotropic hormone, or ACTH for short. These target the adrenal cortex. And again, that makes sense because that's their name, adrenocorticotropic hormone. Whenever you see the term tropic, it means that it leads, it's a hormone that causes another hormone to be released. So the adrenocorticotropic hormone is a hormone that targets the adrenal glands, and guess what it does? Because it's tropic, it causes the adrenal cortex to release cortisol, which is another hormone, hence the tropic in its name. Okay. The 5th hormone that's produced by the anterior pituitary gland is the prolactin, or PRL for short. Prolactin targets mammary glands, and that leads to milk production. Okay. And what else? You've got the 6th one, which is the thyroid stimulating hormone, TSH for short. It targets the thyroid gland. Makes sense because that's in its name, right? Thyroid stimulating hormone. And what does it do? It stimulates the thyroid gland to secrete thyroid hormone. So in that instance, it is what type of hormone?
A trophic hormone?
Beautiful. Exactly. Excuse me. So a test question in the past would be, which of the following is a tropic hormone? So if one of the options was LH or ACTH or TSH, you'd choose that because it's a hormone that causes the release of another hormone. Okay. So that is an example of what you want to do for all of the hormones. Lump them together based on where they're produced, identify what they target, and then, of course, what is their action. Okay, questions?
Oh, I did have a
Yeah.
question.
Oh,
The
uh-huh.
posterior pituitary gland, it does not make any hormones. It only releases them. Is that what it is?
Yes.
Okay. That's why I got that one wrong. Got it.
Mm-hmm. Yep. Oh, you're talking one of the quiz questions, right?
I was
Yeah.
like, I swear there's just two in there, but it doesn't make them. Okay, so that's from the hypothalamus.
Yeah.
Got it. Okay, cool.
Perfect. All right. So with that, I'm going to move into the reproductive system. You have the male and the female, and you already know the answer to this. Which one's more difficult?
Female.
Female.
That's exactly right. So we're going to cover the easier one first, which is the male reproductive system, and then use that to kind of help us understand the female, because female is so much more complicated. All right. So starting with the male reproductive system, you want to be able to know what the functions of the male reproductive system are and describe the male reproductive organs and their functions. And within the organs, I want you to know the testes. And if you know the testes, you should also know the subregions of the testes, which include the seminiferous tubules, the metatestes, and of course, all the cells that are formed in the testes. Beyond the testes, you are responsible for knowing the epididymis, the ductus deferens, the penis, the urethra, and the glands. Understand spermatogenesis in detail, and that's what I'm going to go through next, because if you understand spermatogenesis, it makes learning oogenesis half as easy. Okay? So, and then to end the male reproductive system, you're responsible for knowing the composition of semen and their function. What is the anatomy of a sperm? How do you know when it's mature? How do you know when it's not mature? And so on. Okay? So before we go on to spermatogenesis, there are a couple terms that I want you to be able to know. It's kind of like a reproduction 101. So the first term that you're responsible for knowing is gametes. And gametes is just another term for sex cells, right? So if we're talking about males, what would the gamete be?
Sperm.
Mm-hmm. And then for female?
Eggs.
Eggs.
Yes. What makes a gamete cell a gamete cell? What's unique about a gamete?
What's unique?
It's haploid, and
Yes.
it just has 23 chromosomes.
Yep. It's the only cell in your body that's a haploid. Okay. All the other cells in your body are diploid, which means they all have 46 total chromosomes or 23 pairs of chromosomes. Gametes are the only cells in your body that have half. Makes sense, because what's their purpose? Their purpose is to combine together to become a new cell that represents, yeah, exactly, that represents the combination from the donor cells. Okay?
I have a question. So I just want to make sure
Yeah.
I'm correct when I'm studying. So the
Mhm.
diploid has 23 pairs or 46
Mhm.
altogether, and then for the, how you say it, hiploid,
Haploid? Yep.
haploid, it only has 23. It's not pairs. It's just 23.
That is exactly right.
Okay.
Yep. Mm-hmm. Yep. And actually, that leads to one of the other things I want to talk about, which is chromosomes. So chromosomes contain DNA in the form of genes, right? So in an average cell, in like an everyday cell, except for gametes, you have 46 chromosomes total. Even though there's a total of 46 chromosomes, there's actually just 23 pairs. So only 23 unique chromosomes. And that's for protective reasons. You want to always make sure you have a copy in case something goes wrong. Okay? So when you have 23 pairs, or it's indicated as a 2N, we call that a diploid. So you have a copy of everything. So chromosomes 1 through 22 are completely identical to each other. And because they're identical copies of each other, like chromosome 1, chromosome 1, chromosome 2, chromosome 2, blah, 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 all the way down to chromosome 22 and chromosome 22. So because they are identical to each other, we call them autosomes. Autosomes are when chromosomes are identical copies to each other. The 23rd chromosome pair may or may not be identical because what? Not everyone has the same what?
Sex.
Yes, exactly. So if you're female, you also are identical in that chromosome. So you have an X and an X. If you are male, you only have one X and you have a Y. Where did the X in males come from?
Mom.
Yes. And where did the Y in males come from?
Dad.
Yep. And it's the Y that determines the gender, right? So who determines the gender of a child?
Male.
That's right. Someone should have told Henry VIII that, right? Before he chopped off and he killed all of his wives. But yeah, so it's the males that determine the gender of a child. Okay. All right. Then another term you want to know is fertilization, and that's when the sperm is able to penetrate the egg and combine the chromosomes, resulting in a zygote. So zygote is a new cell that's formed by the combination of the two donor chromosomes. So one donor contributes 23, the other donor contributes the other 23, and a zygote is when you have 46. Okay. Then you have cell division. There are two forms of cell division. There's mitosis and meiosis, or meiosis, whichever way you want to pronounce it, as long as you know what it is. Okay. So in mitosis, it's a cell division. They're both cell divisions, but mitosis specifically is cell division of somatic cells. What are somatic cells?
All the body cells?
Yeah, exactly. They're just all the cells except for gametes. Okay. So they're in one cell that has 46 chromosomes. In mitosis, what happens is it first copies. So it goes from 46 to 92, and it just splits. It divides everything equally, not just the chromosomes, but also the organelles, the fluid. It just splits in half. That's mitosis. And that's why from one cell, you have two new cells that are identical to the original cell. Questions?
Nope.
Okay. So if I were to take your skin cell and your hair cell, are they identical or are they different?
Identical?
Chromosomally, they are identical. That's right. Okay. Yep. So, because why? Because we all came from a zygote, which is one cell that just undergoes millions of mitosis to produce who we are. Some of those cells, even though they're identical, they differentiate. In other words, they pick up a specific function, right? And these cells aggregate together to become the heart, or certain cells aggregate together to become the liver, okay? But chromosomally, they are identical to each other, even though they may
That's
have
so
different
cool.
function, isn't it?
So,
Yeah.
like every single cell, like that's how they can get DNA off of like somebody's toothbrush, right? Because like
Yes.
literally it's got your blueprint right there. on
That's
any
exactly it. You got it. Isn't that wild?
Mm-hmm. That's cool.
Mm-hmm. Yep. Where in meiosis is a form of cell division, but it's different because where in mitosis it's a copy and divide, meiosis has a second step. Okay, so meiosis is essentially mitosis plus more. Okay, so the purpose in meiosis is to reduce the number of chromosomes. Let me say that one more time. The purpose of meiosis is to reduce the number of chromosomes. Why do you want to do that?
That's what I was just about to say. Why do you? I'm so.
Great
Yeah,
minds think alike.
I struggled with this one, so I'm paying attention really hard.
What do y'all think?
So it can meet up with a sperm.
That's it. That's
Oh,
exactly.
it just wants to. So this will form one. No, I'll finish listening.
No, you're right. You're right on the exact right track. So because we want diversity within our chromosomes, right? Because if you have the same chromosomes, mistakes can happen. And then that makes that cell vulnerable and it can die out. And that's why the more diversity you have, the healthier the cell is. Okay? So meiosis is a chromosome reduction so that there's a diversity that can occur when two donor chromosomes come together to become a zygote. And that's why meiosis is specifically to produce gametes. Okay. Would your hair cells undergo meiosis?
No.
No.
No. Would any other cells in your body, except for your sex cells, undergo meiosis?
No.
Exactly.
Because then otherwise your DNA wouldn't be your DNA, right?
That's exactly it. You got it. Yep. So in meiosis, it's a cell division of gametes, not somatic cells, but gametes. And it has two stages. There's meiosis 1 and meiosis 2. So in meiosis 1, you have a division. And already that's different, right? Because what happens in mitosis?
It copies
Copies.
first.
Yeah, it copies first and divides. And that's why from one cell that's 46 chromosomes, it ends up with being two cells that are 46 chromosomes. So they're identical. But in meiosis, your original cell has 46 chromosomes. It's going to produce daughter cells or, the offsprings of that cell, and it only contains half of that chromosome. And that's where the term haploid comes from, because it's half of a diploid. Okay? And that's good. You want that to happen. Okay? So in meiosis 1, it starts off with just pure division. It just splits. It gets it down to the right number of chromosomes so that when reproduction happens, you have a set from one donor and a set from another donor. Okay?
So,
Yep.
Uh,
Go ahead.
So the 23 chromosomes that divides into, it could be a different set of 23
Mm-hmm.
each time.
That's
And
right.
that's why siblings are not exactly the same. Is that?
That's right.
Okay, got it.
Yep. Exactly. So in meiosis 1, it just divides. So that's why if, let's say we're talking about a sperm, okay? So a spermatogonium, which is the stem cell of a sperm, has 46 chromosomes and it just splits. So that spermatogonium, chromosomes 1 to 22 are autosomes. So they're identical copies, chromosome 1, 1, 2, 2, 22, 22. But the 23rd pair is what?
What's the sex? It's the sex
X and
chromosome,
Y.
right?
It is sex chromosome, but which one specifically?
XY.
Yes. So when it splits, one would have the X and the other would have the Y.
Mhm.
Ah, so they're not identical anymore. Okay. So that's what happens in meiosis 1. It splits it in half. In females, when meiosis 1 happens, are they going to be identical to each other or are they going to be different?
Identical.
They're going to be identical to each other. That's right. Mm-hmm. Yep, because the 23rd chromosome they're both what in females?
X's.
Yeah, X and X. Yep. All right, so that's just the first step though. To complicate things further, in meiosis, it has a second step called meiosis 2. In meiosis 2, once it already divides, in other words, it reduces down to two cells that have 23 chromosomes only, it actually is going to undergo a copy and divide. Okay, so those two cells that have 23 chromosomes will have 46 chromosomes temporarily until what happens?
Until it splits again.
That's right.
Okay.
And that's the difference between mitosis and meiosis. So instead of getting 2 cells that are identical, you end up with 4 cells that are not identical. Okay? All right. All right, so let's go. Let's just go through it. That's fine. I was going to skip spermatogonium. I think we should go through it because it gives you what should happen. And then we'll see, we'll go through what actually happens in the female, okay? All right. So spermatogonium, the process of spermatogenesis happens in males, and it starts at puberty. And it goes all the way until a male dies. And that's why from puberty to death, males are producing sperm at a new rate. I'm sorry, not a new rate, but they're producing new sperms throughout the whole period. And that's why males can have children, in their however long that they live, because it's new sperm, right?
It's new sperm, but not necessarily healthy, right?
Not necessarily healthy, but not necessarily unhealthy either.
Okay.
Yep. Where females, the process of oogenesis happens at the female's birth. So during her fetal development. So even before she's born, she's going to develop all the eggs she has in her life. So that means as she ages, her eggs will also age. And that's why a female, as she ages, she tends to have more potential for chromosomal abnormalities during reproduction. Does that make sense?
Mm-hmm.
Yeah.
Mm-hmm. And that's again because she has those eggs during her fetal development. That means that when she's 50, her eggs are also 50 years old.
Yeah. So it's not good for women to sit around and just wait to have kids. It might be better to get it out the way because that's kind of scary.
Yeah. I mean, there are some, cases of women who are, in their 60s, 70s that have children, healthy children, perfectly healthy children, but the odds of that are less.
Yeah.
Yeah. All right. Okay. So normally what happens in spermatogenesis, you start out with a spermatogonium. A spermatogonium is a stem cell. So technically, it's not a gamete. Okay. Which means it's going to have how many total chromosomes?
46.
That's right. And another way of saying 46 is 2n. Okay. Then that spermatogonium undergoes mitosis. And we just learned it, right? If something undergoes mitosis, it's going to produce identical or different cells.
Identical.
That's right. So it sustains the stem cell. So the spermatogonium, which is the stem cell, undergoes mitosis. So it's going to result in 2 cells that are identical to each other. One of them will stay to remain as a stem cell, a spermatogonium, and continue that division process to give an unlimited supply. Okay, where the other one is now going to differentiate. Now you've heard of that term before, especially in bone cells, right? Osteogenic cells become osteoblasts, osteoblast becomes osteocyte. And that differentiation is, think of it as like you have a newborn, okay? And then the newborn becomes a toddler. The toddler becomes a preschool. Preschool eventually becomes a high school. That's differentiation. It's the same cell, but it picks up specific functions. So that high schooler decides that they want to become an OTA, or they decide they want to become a teacher, or that's differentiation. It's still the same cell, but it's become more defined. Okay, so that's what happens to the other spermatogonium. Okay? It differentiates because now it's not going to be a stem cell anymore. It has committed to become a sperm cell. So we call that cell the daughter cell type B. It's still spermatogonium, which means is it a haploid or a diploid?
That's a haploid.
Haploid.
It is a diploid, right? Because it's
Diploid,
still a spermatogonium. It's just now differentiated to become a type B.
Oh, okay.
The only hi,
It's still a
Sorry.
you said?
That's not
it's still diploid. Yep. The
Oh.
only time it becomes a haploid is if it undergoes what cell division?
Meiosis. Meiosis.
Meiosis, exactly. And has it gone under meiosis yet?
Oh yeah, no. Okay.
Not yet. Nope. Don't ever, you don't have to apologize for your children talking. I love hearing kids.
Me too.
See, that voice was so cute. You can just leave them on. Okay. All right. So the type B spermatogome has committed to become a sperm, and that's when it undergoes meiosis 1. Remind me what happens in meiosis 1 again?
It's split. It divides.
That's
Divide.
right.
Divides.
Ah, so now you can call it what?
Haploid.
That's right. So from one primary spermatocyte, it becomes, after meiosis 1, the division, it becomes 2 secondary spermatocytes that are now haploid. Make sense?
Yeah.
Okay, because that's just a pure division. No copy, just a pure division, splits in half. Okay, then in meiosis 2 is actually not, I shouldn't say the same. It's similar to mitosis. The process is similar in that it will copy first and then divide. And that's why it goes from being 2 cells to how many cells total?
4.
4. That's right. Okay. So technically, from one primary spermatocyte, it becomes 4 spermatids. Okay. Those spermatids will eventually, as it's traveling through the ductus deferens, through the epididymis, it's going to develop a flagella, and that's when it becomes a mature sperm cell. Questions?
What was that term? It goes through something to become a mature sperm cell.
Maturation, you mean, or?
Oh yeah.
The spermatids.
Oh, okay. So the spermatids don't have tails or anything.
Right.
Okay.
Oh, I think you're referring to spermiogenesis.
Ah, yes. Yes.
Okay.
Yes.
Yeah. So you want to know the process.
Mhm.
Yep. Mm-hmm. Yep. Okay. So you can see that in males, not only does it start at puberty and goes all the way to death, but it's about quantity over quality. It's about mass production. And that's why probably about 15% of these sperms aren't viable. Okay. Where when you compare it to oogenesis, it's about quality over quantity because every month only how many get released?
1.
Yeah. So it goes through the same process, but instead of keeping what it produces after division, it undergoes what? What's that term? It undergoes what?
Apheresis. Starts with an A.
Yes, it does. You got this. You got this. Starts with an A.
Not apheresis or
So close. Starts with an A, ends in trasia.
atresia.
Yes.
Yeah.
Okay. Yep. So it essentially just dies out. Okay. It's just a way of eliminating the extra chromosomes. Okay. So in females, it's about quality over quantity because it knows that whatever cell it produces, that's going to be the body of the zygote. Because the sperm is just contributing the chromosome, nothing else. Okay.
So basically, the low quality sperm are not going to make it to their target just because, and that's how we keep the bad ones out.
Yes.
Okay.
Yep. Yeah. I'm laughing because there's an animation that was made based on that. So it's funny. If I find it, I'll email it to everyone. Okay. So let's go through oogenesis. Oogenesis is wicked hard. Okay, because the reason why it's hard is because you have to follow 3 cycles that occur at the same time, simultaneously. You have to cover oogenesis, follicular genesis, and the changes that happen to the uterine lining. That's what makes it so complicated. Okay. So here is the, nope, that's not it. Do I not have it in here? Oh, there we go. Okay. I'm like, I disappointed myself. Here you have the full picture. And if you can understand and describe this picture, you're going to do fine. This picture is so hard. I don't even know how to tell you. I mean, it takes a lot to be able to understand this picture. So if you're struggling, give yourself grace, give yourself time, do 1 cycle at a time. Okay. So when you're studying this, I would highly recommend studying just the ovarian cycle or just the uterine cycle. And then you can combine them together once you understand each one individually first. And that's why hopefully all of you have found the PDF, because I created, it's one of my proudest, work that I've done for this class is to create that PDF for you guys that shows the cycles and the days by the, all the 3 cycles by the days.
Mm-hmm.
Do you know what I'm talking about? That PDF?
Yeah.
Okay.
Yep, that's super helpful.
Yes. Use that to study. Okay. All right. So we're running out of time. Any questions before we end the session?
Let's see. Okay, just one, I think. So after the secondary follicle, there was a tertiary follicle, or that's just like basically the step. The first one that's mature basically shuts down the continuing development of any other egg, right? So only one egg is going to be expelled from.
Perfect.
Okay.
Yep. Yeah. And that's another thing. In females, about 20 to 25 eggs actually get stimulated to mature.
Mm-hmm.
Of those 20 to 25, only 1 gets released. And that's because the healthiest one that matures the fastest creates a domino effect that causes the others to just kind of die out. Mm-hmm.
Except in twins, which is bizarre.
Well, twins actually could happen when 2 eggs are released from the different ovaries. So it's
from 2 different ovaries. Oh,
yes.
okay, I was wondering about that.
Yep. Normally 1 ovary is active per month, and then they kind of go back and forth.
Okay.
Mm-hmm. The other way twins can form is that a zygote is made, and then that zygote splits completely.
Yeah, that happened to me. I mean, my sons.
Yes.
Okay,
Yep.
cool.
In which case you have identical twins. Yep. All right. So, yeah, I'm just going to spend a few more minutes if that's okay. So in oogenesis, it starts out as oogonia, so very similar to what we covered in spermatogenesis, right? So that's a form of mitosis. And that's why during fetal development, it's able to repeat itself. So this one will kind of be able to go back and be that constant stem cell during fetal development. Then the one that differentiates or has committed to becoming an egg will then be called the primary oocyte. Once it becomes the primary oocyte, it starts meiosis, but it never completes it. So right here. Okay, so it starts meiosis 1, but never completes it. So it goes under cell arrest until puberty hits again. At puberty, that's when it's going to finish meiosis 1. And even when it finishes meiosis 1, it's only going to keep one of its divisions. The other one becomes a polar body, and it's just a way to reduce the number of chromosomes. So you can see where in males, they keep all of their divisions, right? In females, it's just a way to reduce the chromosomes. Okay. And it only goes through meiosis 2, if fertilization happens. If fertilization doesn't happen, it just gets expelled during menstruation. Okay. All right. So if you want a more detailed explanation of that, don't forget I do have the videos that I recorded for my classes. Make sure you take a look at those. Okay. Any questions before we do some quiz questions?
No.
Okay. All right.